Yes. Now people are popping up. And okay, but I mean, it's 49 still, I think, but you can start whenever you want.
Okay, hello everyone. Now we are. I have my the second talk, which is about immunostaining troubleshooting. Okay, why it's not working?
I can see your screen.
Okay.
Mm.
So I divided this lecture in three parts. And in the first part, we will talk about a little bit about the differences between immunohistochemistry, immunocytochemistry, and immunofluorescence, different types of antibodies, and when to choose chromogenic versus fluorescence staining. In my second part, we will talk about where to find our antibodies and what antibody validation means. And in the third part, we will focus on the certain immunostaining steps. I will try to give you some tips and what to pay attention to. So let's get started. In immunostaining, we quite often encounter the abbreviation IF, IHC, ICC. So what is the difference? The I on all of these abbreviation come from the word immuno, which means that the assays are an antibody-based method to detect a specific protein. The H in AHC comes from the word histo, which means that we will work with tissues or 3D tissue models. The C in the ACC, the first C comes from the word cyto, meaning that we are working with cells in monolayer or suspension of single cells. The chemistry, the C from in ICC and HC and ICC comes from the word chemistry, meaning that we detect the signal using an enzymatic reaction. And the F comes from fluorescence, meaning that the detection of the signal, we detect the signals using fluorescence. And this particular it's regardless whether the sample is a tissue or a cell monolayer. So now we have a little idea what all these abbreviations actually come from and mean. So a few words about immunostaining, just a very quick recapitulation. So immunostaining is an antibody-based method for detecting the protein of your interest. And the antibodies are macromolecules, which are naturally produced by the immune system of the animal, us, to defend ourselves against foreign matter, which called antigen, that do not belong to them. So the antigen is the protein that is recognized by the antibody. Here I depicted the protein, the antigen, coming from the human. And then we have our antibody, in this case IgG, which comes from a rabbit. The antibodies come in different flavor called isotopes. The mammals produce 5 different antibody isotopes: IgG, IgA, IgM, IgE, and IgD. And 2 of them, IgG and IgA, had also several subclasses. And always the IgG, in the case, for example, of IgG, the IgG is the class and the 2A is the subclass. So how should we detect? And before we go there, this antibody, the part that actually binds to the specific antigen is called the FAB fragment. It comes from fragment antigen binding, and this antigen specific. And the part The other part, the FC fragment, comes from the abbreviation comes from fragment crystallizable region, and is species specific. So how do we detect our protein? That's so we can do a so-called direct labeling. Then we label our antibody with either with a fluorophore or a chemical, and then we can detect it at the microscope. We can also perform indirect labeling, and in this case, again, we have our antigen, which is recognized by our antibody, which comes from a rabbit. And now the FC part of the rabbit antibody is injected into a different host, let's say a mouse, and the mouse will produce its own antibodies, in which the FAB part of the mouse antibody would recognize the FC part of the rabbit antibody. And now we will label the FC part of the mouse antibody with our either enzyme or the fluorophore of our choice. The part that directly binds to our antigen is called primary antibody, and the antibody that will recognize our primary antibody is called secondary antibody. So now we know how this works. There are many types of antibodies. Let's go through them a little bit and see what are the advantages and disadvantages using them. We have monoclonal antibodies, which are produced by injecting a host animal, and it can be rabbit, sheep, goat, chicken, alpacas, for example, with the antigen that will give rise to an immune response, and the bodies will be extracted from the animal blood. What we extract and what we then we will get will be a mixture of antibodies, all recognizing different epitopes but of the same antigen. They have a high antibody affinity against the antigen due to the fact that they can recognize multiple epitopes, which also make them more sensitive to detect low quantities of the antigen. But on the other hand, will also have high chance for cross-reactivity, exactly to the fact that they recognize multiple epitopes. And it's another negative part regarding polyclonal antibody is the relative high batch variability. The monoclonal antibodies are generated in vitro by hybridoma B cells from mice. They recognize only one or only a single epitope on the antigen, which gives them high specificity due to the fact that they recognize only a single epitope, and also it means low cross-reactivity. We have a low batch-to-batch variability between when they are using monoclonal antibodies, but they take time to be produced, and they are more expensive. We also have recombinant antibodies, which are monoclonal antibodies produced in vitro when we are cloning the different parts of the antibody, the heavy and the light chain in expression hosts. Again, they only recognize a single epitope on the antigen, and they provide an unlimited supply of identical antibodies, so thus they will remove the batch-to-batch variability. We also have other type of antibodies, so-called antibody fragments, which they can be produced if we, for example, we have the IgG antibody, and we incubate them with different enzyme, for example, papain, then we will get two monovalent FAB fragments, each of them with one antigen binding site. If we incubate the IgG antibody with pepsin, for example, then we will get a so-called FAB2 fragment, which has two antigen binding sites. What are we using these antibody fragments for? Well, they are highly used to block cross-reactivity. Then we are using primary antibody raised in the same host species. or if you want to block the binding of your secondary antibody to Fc receptor of certain cell types, for example, B cells or NK cells. You can also use them as labeled secondaries, especially the FEB2 fragments. And the advantage of using these antibody fragments is that they penetrate the tissue more efficiently due to their smaller size. But you will get a lower signal since fewer dyes or enzyme can be conjugated to the fragment. Another type of antibodies are camellids or nanobodies. And these camellids, they were originally identified in camel. That's ergo where the name comes from. But they can also be found in alpacas, llamas, and some shark families. They only contain, as you can see, the heavy chain, so they don't have any light chain. And this variable part of the antibody, it's very small, can be isolated, and this is why they are called nanobodies, and they can be cloned. So why you should use the nanobodies? First of all, because of their small size, they can penetrate the tissue more efficiently. They are more stable at room temperature than the standard IgGs. They have high affinity and specificity for their target antigens, and they can easily be tagged with fluorescent protein. For example, if you want to do live imaging or other fluorophores. Another type of antibody are so-called single-chain fragment variables. They only contain the variable heavy and the light chain. And of the IgG antibody, they are recombinantly produced by combining the genes for the variable chain and variable light chain, and they are coupled with a linker. Again, due to their small size, they easily penetrate the tissues, and due to the lack of the Fc region, they give a low background signal. They can also easily be fused to different fluorescent proteins or enzyme-like proteins or affinity tags. So it can be detected easily in different combination. The background of using this single chain fragment variable is that they have lower binding affinity than the standard IgGs. So now we know the different type of antibody. We choose what we would like want to use as an antibody, primary or monoclonal or polyclonal or nanobodies. But let's see what is the difference on the technique that we are using. What's the difference if we are performing chromogenic versus fluorescence detection? If you want to detect our antibody via chromogenic or fluorescent detection method. The chromogenic labeling is a chemistry-based method, and the fluorescent labeling is a fluorescence-based method. In both cases, you can either directly label your antibody, or you can perform indirect labeling. So when we are performing chromogenic labeling, what happens? We have our antibody that will bind to our antigen, and then our, for example, if I do an indirect labeling, our secondary antibody will have an enzyme bound to it. After the primary secondary antibody, we perform the reaction, we can add a substrate, and we will get a precipitate where we have the primary and secondary pair together. And quite easy. Usually, this is a kind of image you get, then you look in the microscope. You can see where the precipitate is. If we are performing fluorescent labeling, then of course again, we have our antibodies, and which will recognize our antigen, and our secondary antibody. To our secondary antibody, we bind fluorophore that absorbs and emits light, and then we can detect it also under the microscope. So when should we choose what? We have a chromogenic labeling or fluorescent labeling. When we perform chromogenic labeling to visualize our labeling, we need a color cameras which are less sensitive than the black and white cameras which we use when we are performing fluorescent labeling. The dyes, we can use dyes with different color, but only if we don't have overlapping antigens, meaning that the localization of our antigens should not be on overlap at all. In the case of fluorescent labeling, it's possible to detect more molecules at once, even with overlapping antigen localization. The contrast, sharpness, and resolution are lower in chromogenic labeling. In fluorescent labeling, we have always a better contrast and better resolution. Autofluorescence is not an issue when we perform chromogenic labeling, but sometimes the tissues can have themselves can have some color. When we perform fluorescent labeling, autofluorescent, it is a problem. No bleed-through issues, and the label is preserved for a longer time than when we perform chromogenic labeling. In fluorescence labeling, we do have a risk for bleed-through between fluorescent dyes, and the fluorescent dye can fade both during imaging and storage. So choose the appropriate detection method depending on your scientific question and and the resolution that you want to that you need to answer your scientific question. Let's move on to our second part of the talk. And now that we know what kind of antibody we want, I want to have a monoclonal, or I need a polyclonal antibody. Let's see how we find them and how we can see if get an idea the antibody will perform well for our experiment. But just before we move on, how many of you have checked if the antibody you use is validated? Can you raise your hands? Then you look for antibodies. How many of you actually check?
Good. I don't know what validated means.
So validated, it means that the manufacturer actually looks and see that your antibody is working for the application, for example, that you need the antibody for. That's good. That means that all of you are aware of how important the validation is. Good. That will make my speech easier. So, what is a good antibody? A good antibody should fulfill the following criteria. Okay. Specificity, which is the most critical issue, so it should only detect the target antigen. Sensitivity, it should detect low amounts of the antigen. And reproducibility, the same level of specificity and sensitivity across batches and time. That's what we want, the requirements for a good antibody. Now, where do we find our antibodies? Well, most of the time we are asking around, we are looking at publications at the vendor side, but this takes a lot of time, and you need to take your time to find and to choose the right antibody. And keep in mind that 50% of the commercial available antibodies are either not working or actually they might not fit the purpose of your experiment. So when you start, you go to antibody citation, scientific publication, and these are generally giving a very good overview about the available antibody and what is being used by the other researcher. But keep in mind that just because an antibody was cited by many journals does not necessarily mean that it is the best for your application. And you should be very critical and look carefully at the images that they provide in publication, the protocol that they use, and the experimental setup before deciding, oh, this is actually, I will use also this antibody. And as I mentioned, finding the right antibody is very time consuming and sometimes error prone. So what could you do? Well, a good suggestion from our suggestion is to start to search the databases, first of all, those with validated antibodies. And one such a database is the Human Protein Atlas, which it's an open source database with thousands of antibody against human proteins. And they all contain high resolution images, both immunohistochemistry and immunofluorescence images for all the antibodies which they have, because they are all validated. Another database highly recommendable is the Antibodypedia, which is also a searchable database of publicly available antibodies where they have validation score for the antibodies as for their various applications. So this is really a very good start in your search for antibodies. There are also other more specialized antibody search engines, also containing comprehensive databases, sometimes with millions of antibodies, where you can search for the antibody of your interest by application and so on. And going to all these searching engines would spare you a lot of time. And on top of them, these databases also put together many vendors, many manufacturers of antibody in one place. So you also can read comments from other researchers. You get tips for how to for different antibodies, and also a list of publication, direct list of publication where certain antibodies were used. So you really spare quite a lot of time if you refine your search than using this antibody search engine. Here I only name up a few of them. There are pretty much many more, but these are the most common and reliable antibody search engines. So now that you spend some time and you looked and you find maybe the antibody of your choice, you said, yeah, this will work for my sample or for my protein, then of course, the first thing you will do, you will click on and you will look to the data sheet. And the question is, how much information should you find about this antibody on the data sheet? Because a nearly empty data sheet is a strong recommendation or indication of a non-validated antibody. So there are a few things that are a must, and they should be on the data sheet. Identification details, storage and handling, and validation data. What should the minimum information included in the identification details are, of course, clone number or the commercial product number, antibody ID or RID number. So you can always go back and look up or search about this particular antibody. Of course, the type of antibody, to know exactly if it's a polyclonal, monoclonal, or recombinant nanobodies, the isotype class and subclass, because this will, of course, is a necessary information for the next step in your immunostaining. It's a clear definition of the immunogen. How was this antibody erased against? The peptide, a small peptide, or a protein fragment, or maybe the full-length protein. Because if you cannot, if you don't know this information, it might be much harder to decide if the antibody is specific enough or not for your application. Of course, specificity and species reactivity, to know the target protein and the epitope, and the species which has been shown to react, human, mouse, rat, you name it. And probably one of the most important information, the application thing. You are interested in immunostaining. So if this information is not there, or is of a very poor quality, then it will already give you a hint that, yeah, well, you might need to validate the antibody yourself, that it works actually for the application you want, immunostaining. Storage and handling, concentration of the antibodies and additives and the buffer. This information is crucial. First of all, it will help. If it's not there, you spend a lot of time to optimize your protocol. The additives and the buffer, if the information is not there, it might prevent you from using this antibody. For example, if you want to directly conjugate it to different fluorophores. The storage is a must, considering how expensive these antibodies are. So it's quite important to know how to storage them, and of course the expected lifespan of the antibody when stored correctly. And I think the third information that is important, the validation data. They should provide a list of experimental methods they use to validate the antibody. For example, how did they validate the antibody? What kind of method did they use? Would be Western blot. Fluorescence, histochemistry, IP, flow cytometry, which method they use, what type of sample they use to validate the antibody. You might, you work in a tissue, they might only validate the antibody in cell lines. Then you already have a question mark, where will it work in my sample? Do they perform appropriate controls? Do they give any, do they recommend any dilution? And first of all, like in our case, they have to show some representative images. And here I just have a screenshot. I went to the Human Protein Atlas homepage, and I was looking how do they validate their antibody. And I just randomly choose some the most commonly used sources to buy antibodies. And I actually looked and tried to find the information, how do they validate their antibody. And you can see most of the most serious companies and manufacturers, they do write it very clearly. This is how I validate the antibody. So you have an idea, and you know you have a good starting point. So when should you as a researcher validate your antibody? Well, you should do that if the vendor did not validate, you don't find any information about it, especially for your type of sample. As I said, if they only validated in cells and you have tissues, yeah, well, then it's time you have to do the job. If the vendor did not provide extensive validation data or data specific for your application, maybe they validated the antibody for a flow cytometry, but that doesn't mean that the antibody will work whatsoever for immunostaining. You find you have unsatisfactory validation data, or the publication that is the citation that the manufacturers provide. Just because it's recommended for immunostaining, for example, but you don't see any image whatsoever, that it's a sign that the antibody actually was not tested for immunostaining. When you produce your own antibodies, you also you should definitely, of course, validate the antibody. There are several antibody validation guidelines, and I highly recommend. I put some of them on Canvas, and I highly recommend the paper from 2016 from Perulin, where they take you through all the steps required for a good validation of your for an antibody. But there are a few more there that will definitely give you a guide, will give you guidelines and ideas on how to perform the validation for your antibody. And so what will you look for when validating your antibody? You should look for specificity, reproducibility, and sensitivity. How you will check specificity. You can use different either genetic method. The most used methods are the genetic methods. Then you will have gene knockouts or knockdowns, and then you will look for the signal. So you should have no signal, proving that your antibody only binds to the intended target. Western blot is highly used to proof specificity. You should only get one band, and we'll come back a little bit to the Western blot. Or you can have expression controls, positive and negative controls. Basically, if you work with cell line, it's very easy because you can, or tissues too, for that matter. Then you can use cell lines or tissue node to express your target protein, and that will be your positive control, or those that do not express negative control. Then you look to the reproducibility. What you want to know is to confirm that the antibody performs consistently across different experimental runs and antibody lots. And when it comes to sensitivity, titration, you need to determine the dilution for the primary antibody, and of course later also for the secondary, that will give you the best signal to background ratio and signal to noise ratio. Just a few words about Western blot. If you use this method to prove the specificity of your antibody, it's one of the easy and cheapest to perform, and it's widely used to determine antibody specificity. But the Western blot will only inform you that the antibody is able to bind to the denatured protein. So if your antibody was raised again, the folded protein, the antibody might not, the Western blot might not, cannot be used. You always need to perform the Western blot using extracts of your own sample, example, tissue extract if you work with tissues. Always include a positive and negative controls, because the proper design of these controls are the key to confirm the specificity of the antibody. If you only get one, if you get one band at the correct molecular weight, that will confirm that the antibodies bind specifically to the protein of your expected molecular weight. But this it's only valid if you have the correct controls. If you have you get several bands, that could also mean that the protein is due to the different post-transactional modification, for example. This is when you have to understand the biology behind your target to be able to interpret the data. And if your protein is very have low expression in your tissue or in the cells, actually you might have problem of detecting it with the Western. So you have to move on to a different method. Now let's talk a little bit about reproducibility between the experiments, and we can look at the reproducibility from two angles. First of all, the reproducibility between the experiments and between batches. How you you confirm the reproducibility of an antibody. If you work with a current batch, then what you can do is to run the same immunostaining protocol on for example, on 3 different days, and compare that you get similar results. But how about reproducibility into the batches? Because sooner or later you will run out of the batch of the antibody that you work with, actually. So first of all, you have to think ahead and to order a new batch well before the first runs out, so you can compare. Do I get the same results with my new batch of antibody? As I already said before, the polyclonal antibody have a higher risk to batch variability. So if you know that your experiment will run for a very long time, you maybe want to think twice to use a polyclonal antibody. Also, that polyclonal antibody are not the best choice if your application has a very low tolerance for variability. Since we know that we do have variabilities if we use polyclonal antibodies. But if only the polyclonal antibody is available, then you can also ask for the same lot. Reproducibility, you have a lower risk if you use monoclonal antibodies, but then you might have encounter other challenges. For example, if you want to look for low abundant proteins or proteins with epitope variability in the sense of post-translational modification or conformational changes. The recombinant antibodies are probably the best because they show less variability between batches, but they are quite expensive. Always contact the company and actually complain if, for example, the new batch does not perform obviously as well as the previous one, or you need to increase or to change far too much things on your protocol to actually get the same signal. Of course, the reproducibility will also be affected if the antibody is not stored correspondingly, because of course antibody also degrade in time, and they won't work as expected. So always store your antibodies according to the manufacturer's instructions. Now, just a few words about the stringency. I think that you heard about this word before, stringent conditions. So what do we do? Then we look at our sample and the microscope, and we see very low signal and some dots outside. What should you do? What should you do, and what is the first thing that most of us are doing? The first reaction is to increase the antibody concentration, but because then we think that, oh, we will get a better signal. But actually, this is not this approach will backfire quite a lot, because with this we'll increase also the specific staining, and we will decrease the contrast. So what should we do? Well, as we already heard before in Sylvie's lecture, we should apply some stringent conditions. And by stringent conditions, we mean that we make it difficult for the antibody to bind. And what do we mean with stringent condition? That we decrease drastically the primary antibody concentration, we incubate it at 4 degrees, and to compensate with the low temperature, we will incubate it for much longer time, usually overnight in the cold room. By doing this, we will assure a more specific signal, and will improve the contrast between the signal and our specific background. And of course, anyway, then you are using antibodies for the first time, you should make a dilution series, and you go with big steps to actually find what is the best dilution. So we can improve our signal first at the antibody side by providing the stringent conditions, but we can also improve the detection and at the microscope. We can improve the sensitivity of the detection during imaging, also by making sure that we have a good efficiency of excitation and fluorescent detection, and that we don't have, for example, autofluorescent in our mounting medium. So the first impulse that we want to do if we have a low signal, put more antibody. No, don't do that. Make new samples and make dilution series or use the stringent conditions of low temperature, 4 degrees, and longer incubation times. Another piece of advice is is to be aware that you don't actually buy the same antibody twice. And this is happened, unfortunately, far too often. So what you buy an antibody and it might not perform or work well, or as you expected. So what you do, you go again and you look and you try to find another antibody. But quite often, it happened, we had users who had this problem, they actually bought the same antibody twice. So how do you know that you actually buy the same antibody? You have to do a little bit of a detective work. So you should, because you don't want to waste time testing actually the same antibody twice and pay a lot of money. So what are the signs that you are dealing with the same antibody? For example, look at the formulation in the data sheet. It is exactly the same. Yeah, well, you should put a question mark into your head. If you see the same Western blot or the immunoprecipitation or the same immunostaining images, that something cannot be. So you compare with your old data sheet with the data sheet of your new antibody. How can it be? How are they exactly the same? That's something it cannot be, isn't it? If it's a different antibody. If you have the same vial size, quantity, and dilution, if they recommend exactly the same protocol and the same reference list, that means that the antibody is that you actually are looking to the exact same antibody. It was just the name that was changed or the lot number, because yeah, of course, different vendor, they might use their own labeling. Just that the price is different doesn't indicate at all that is a different antibody. Always buy and work with companies that will work for you and will help you to troubleshoot your antibody, especially if it doesn't perform as expected. So in conclusion, always select the best antibody of your application, best antibody for your application. So use the search engine to find antibodies. It will go much faster, and you will also on top of that gain a lot of information. If you look at the publication and you decide, oh, this antibody would work for me too, be very critical at the data that is presented. Only look for antibodies with complete data sheet that have identification details, storage, handling, and validation data. And again, look for antibody that passes rigorous data validation for your application. Because if you don't find an antibody that was validated and you still want to buy it, you have to do the validation yourself. Always check the reproducibility between batches. And if you have to buy a new batch of the same antibody, order it in due time so you can compare. and purchase the antibody from vendors that are willing to help you with optimization and validation of your application. And again, pay attention so you don't buy or test the same antibody twice. So let's have a break. If anyone has questions, 10 minutes break. I'm happy to answer.
So 10 minutes breaks means we come back at 45 past?
Yeah.
Or 44, yeah? Are there any
45 is okay.
questions? Any questions for this first part? Or reflections for your own experiments? Well, we're staying here, so in case you have a question, you can ask, and otherwise we can have a break.
Okay, so let's get started with the last part of my talk, and which will focus on actually the immunostaining steps. So what is the difference between there is any difference between staining cells or tissues? Because in the immunohistochemistry, immunocytochemistry, they do make a difference. Then they in their abbreviation, if it's immunohistochemistry, it's about tissues, immunocytochemistry cells. But then we are talking about we include also immunofluorescence. Is there actually any difference between them? But the only difference actually between them, the difference lies in the steps required before the immunostaining. So then we are working with cells, they are monolayers or in suspension and very thin samples. And all we have to do before performing the immunostaining is fixation. It's a fast and easy method, and then we can move on to perform the immunostaining step themselves. Then it comes to tissues. The tissues are thicker, and additional steps are required before immunostaining. Depending on the amount of material that we have and what is our scientific question, we can either work with whole mounts or with infections. If we are working with wall mount, we will fix our samples, and afterward, or either before or after, we will have to perform clearing to make the sample transparent. And thanks to them, they are very. And after that, we will image our sample, and then we are imaging wall mounts. We get a very good 3D info of our sample. But then it comes to thin section. Well, we can treat them in different ways. Either we can freeze them, and the so-called cryosections, or without freezing them. If we are freezing, either we perform fresh freezing, and in that case, first we embed them in OCT, and they are flash freezing them in isopentam, which is cooled in liquid nitrogen, and after which we section, and we are fixing them. And it's a very fast method, but it can give rise to some tissue damage. The good thing is that we don't need to perform antigen retrieval. The other way is to fix them first and then freeze them. So we fix our tissue, and then we are immersing it in sucrose, which is used as a cryoprotectant. After which we are embedding them in OCT, and we are slowly freezing in isopentane or dry ice. And only after that we will section them. So already with the cryosection, we can treat them into different ways. This second option, it's much slower. It also gives, but it's more gentle to the tissue, but we can still get some tissue damage, and antigen retrieval it's needed. Or we can decide we don't perform any freezing, but what we have to do is to first we'll fix our tissues, after which we will embed them in paraffin for the tissues, or low melting point agarose for the 3D tissue models. And only after that we will section them. So if we choose to handle our tissues in this way, we have to count that paraffin embedding. It's much more. It's a much more slower method, but it preserves the tissue structure very well. Unfortunately, there are fluorescence proteins that are affected by this method, and it will always give rise to autofluorescence. And on top of that, antigen retrieval is needed. So when we talk about the immunostaining, depending on the type of sample that we have, And then, of course, our scientific question, we will treat our material in a different way, till the different way. But the only difference that actually is between all these methods, it lies in what we do before the immunostaining, how we perform the fixation step, and a few steps that we need to perform to be able to get our samples. When we are which are the controls that we need to perform, then we are performing immunofluorescence, because controls are a must. They are the key requirement for a robust data interpretation. And you should always have controls for each new antibody that you are using. And the controls samples should be processed in exactly the same way as your main sample. So you are running them in parallel next to each other. So one of the controls that it's a must, it's a no primary control. What does that mean? No primary, only secondary antibodies. And this control is needed to determine if your secondary antibody is binding non-specific to your cellular components. So it's an important control, and you should not get any signal. But if you get a signal, let's say, for example, bright dots on your sample or outside, which have they look like the breeze. That actually can come from your secondary antibody aggregate, which actually form due to the fact that you didn't handle your antibody correspondingly, or your storage was not proper. So what you can do, for example, to get rid of these dots or debris or that aggregates, is that first you mix, you vortex your tube very well, and after that you give it a very quick spin. And then you take your secondary antibody, you only take from the supernatant. So then you will get rid of these very big blobs of antibody lingering around. Another important control is the autofluorescence control, and we already talked and we've heard about it, which means no primary and no secondary antibodies. But you should treat this sample exactly like your main sample, fixation, blocking, and all the steps, just that you omit the antibodies from all the solutions. And this is needed to determine if you actually have autofluorescence in your sample. Again, you should get no signal when visualizing the sample. And if you get a signal, that means that your sample is autofluorescent and you have to do something about it. Please don't ignore if you might actually don't have a specific use of this autofluorescence in some way. Of the isotype control, when should you use them, and what is an isotype control? This antibody is basically, it's exactly like your primary antibody, the same class and subclass, but it's not able to bind to your antigen. And when should you use or perform an isotype control? And if the tissue is involved in immunity, which means that it might have FC cells that present the FC receptors, or if the tissue is infiltrated with immune cells, or if your primary antibody is raised within the same species as your tissues. Your tissue that you are working with. For example, mouse anti-Mars and human antibody, which are extracted from blood, for example. And again, no signal should be detected. Now that we know we have our sample, we cut our sections, we know how many sections we have to cut, because we know how many controls and which controls we have to have parallel with our sample. It's time to start with the immunostaining. About the fixation, we talked in our previous lecture, the requirement for the fixative, and our sample was in one way or the other past that first step of fixation. What is the permeabilization used for? It makes basically hole in membrane, so it allows the antibody penetration, and there are different type of reagents, and the choice of them will make a difference if your antibody can reach the target or not. There are two main categories: the detergents or the organic organic solvents. In this category of detergent, the mostly used are saponin, digitonin, and twin-20, which are considered mild detergents, and they will permeabilize only membrane containing cholesterol. So if you want to reach the nucleus, this detergent will not work for you. And on top of that, the detergent has to be included in all solution during the entire labeling procedure. But we also have Triton X100 on MB40, and these are more harsh detergents, and they will permeabilize all membranes. In the family of organic solvents, we have methanol and acetone. These are mostly used, but and they will permeabilize all membranes and dissolve the lipids, and they make quite big holes in the membrane. Then you use them. You should always perform the permeabilization on ice, and you have to keep in mind that it may damage the microtubules or other organelles due to the harsh action on the cells. So you always have to use the permeabilization agent that is suitable for your scientific question. If you use the wrong one, it might remove your target. Also remember that methanol and acetone are also used as fixatives. So it doesn't really make sense if you fix some samples with PFA that you then use methanol as a permeabilization agent if it's not in particularly specified in your protocol for some reason. And always adjust the concentration and time depending on the thickness of your tissue. If you are using these organic solvents for permeabilization or fixation for that matter, always be sure that your antibodies are compatible with it. So antigen retrieval, it's also part of the immunostaining, especially in paraffin embedded section or in cryo section when we are performing the fixation after we embed it in OCT. And what is the role of, why do we perform antigen retrieval? Because we have to break the crosslinks that were generated by fixation, and by this unmask the epitopes. And it's always done before you start the staining, the immunostaining, and you add your primary antibody. And there are two main methods to perform antigen retrieval: heat-induced antigen retrieval, and as the name says, it requires high temperature. This means that some tissue, especially those who don't attach very well, even in the super frost, very highly adhesive slides or cover slip, they attach they might detach due to this antigen retrieval method. And one of the tips we have, if you can, and your setup allows you, instead of having your sample vertically facing down, vertically, that you might maybe you are able to incubate them horizontally in the buffer that you use to perform the antigen retrieval. There are different buffers. The most common are 3-CDTA and citride buffer, and with different pHs. This is and I say different pH because even within these buffers, you should adjust the pH that works best for your sample and your antibody. And I will show an example later. It's a method that requires quite a bit of optimization. As I said, it's a method that is sensitive to the pH, time, temperature, and the buffer composition. And you need to play a little bit around with all these parameters to get the best antigen retrieval result. And here, as I said, it's an example where you can see you have tissue sections, and there are two different antibodies, and they try different buffers and change the pH between them. And it's very clear that, for example, for the progesterone receptor, the glycine buffer, which is with a very low pH, works very well. But already with a TRIS buffer, a slight difference in the pH from 9 to 10.5 totally altered the binding of the antibody. In the same with kedarine, neither of the previous, the citrate glycine buffer don't work, and within the TRIS buffer only with a very high pH. So playing a little bit with the pH, it can make a very huge difference in your for your antibody, and to unmask the epitopes so your antibody can bind. So here, the heat-induced antigen retrieval is not only about heat, that you have a high temperature, but it's also about the pH and the ionic strength. And I can highly recommend this paper, which very nicely takes you through all the steps and why it's important and how to perform in the best way this antigen retrieval step. The other method is so-called enzyme-induced antigen retrieval, where you can use different enzymes, proteinase K or trypsin or pepsin, that will cleave the peptide that are masking the epitopes. But as the previous one, optimization is required for both for the enzyme concentration, but also the length of the treatment. And this method it's more prone to destroying the tissue, and that's the antigen of interest. Can you avoid antigen retrieval? Well, you can at least give it a try. You can first start by diluting the antibody at different pH or cation concentrations. You can use more stringent condition for when you incubate your antibodies. You might choose to choose a polyclonal antibody instead of a monoclonal, that's giving the chance of maybe different epitopes are not masked, so the antibody can bind, or you can use nanobodies. Now we move to the next step in the procedure and blocking. What do we do blocking? We need to block and to perform a blocking step is so that we are blocking and with competing with the specific binding of the antibodies. And it's performed for all type of immunostaining, for immunohistochemistry, immunocytochemistry, and immunofluorescent experiments. And of course, it's done before adding the primary antibody. And there are many blocking reagents. The most commonly used is normal serum, and that should the serum should be from the host of your labeled antibody. If you have secondary antibodies from different species, it might be necessary to mix the blocking sera from all the species of all the secondary antibodies. You can also use protein buffers, which are concentrated protein solution. The most commonly used are BSA, non-fat milk, casein, and gelatin. And you have to, then it comes to BSA, there are different concentrations, depends on the protocol you are following and your antibody. but it's very important to keep in mind to order the correct BSA, which is free of endogenous IgGs. So to avoid a cross reaction with secondary antibody, which of course it will also will give you, so to avoid the risk for high background. You can also use non-dried fat milk. This is also usually at 1-2% concentration. But this particular blocking buffer is not recommended. Then you are looking and you want to detect phosphorylated proteins, or you want to perform assays that include a biotin binding site. And all these buffers, you should have twin 20. That's why most of the these proteins are diluted in so-called either PBS-T or TBS-T, and with T stands for twin 20, and should be always included in the protein buffer, but much lower concentration than when it used as a permeabilizing agent, and this is to prevent unspecific binding. FAB fragments can also be used, and as I mentioned already in the first part of my talk, and they are actually highly used to block endogenous immunoglobulins in tissues or on the cell surface. For example, when the primary antibody is raised in the same species as the sample. And here I give you an example. We have a mouse tissue, and we have some endogenous IgG, probably a cell presenting for, let's say, an Fc receptor or something. And we want to use a mouse antigen X. So it's mouse antibody and mouse tissue. So the first thing that we will do, you will take your FAB fragment, and this will be your blocking. So you can block the sites on the endogenous IgG, and only after that you had your primary antibody, that the one that you want to use to detect your antigen X, after which you can also add your secondary antibody. So it's quite used quite a lot. So also the take-home message, always try different blocking agents to monitor both background, negative control, and the signal, the positive control. So choose the best that will give you the best signal to background ratio. Then you use the blocking buffer for dilution. Diluting your antibody should be the same blocking buffer that you use to block your sample to ensure the most optimal assay conditions. Always follow the instruction provided by the vendor that sells the antibody or the kit. They will tell you exactly what is optimal for that particular antibody to be used with. And the last part, now we came to washes and why we are performing them. And I would say this is one of the part that a lot of people, try to cut corners. Yeah, I already washed it twice. It should be enough. Actually, it's a very important step. And if the goal is to, of course, primary goal is to remove onboard antibody and reagents. And by that, it will reduce your background. So it will improve your signal to background ratio, will remove weakly bound antibodies, to improve the specificity. And of course, it will ensure reagent efficacy, because so the next reagent used in the protocols will work correctly. If you don't wash properly between different reagents, the next one might not perform at its best. But always be gentle, especially when working with cells. And here is an example. Then harsh pipetting between washes actually remove quite a lot of cells from the center of the well, and also from the well of the multi-well plate. So please be gentle when you are washing. Do not omit the washing step or reduce the incubation time between washes. There is a reason why they are in the protocol, and the manufacturer always has they are mentioning a certain washing step with a certain amount of time between. And that is the most common buffers are again PBST or TBST. T is important, not to be omitted. Don't be in a hurry, make the correct solutions. TBST, you should use the TBST as a washing buffer if you perform immunostaining of phosphorylated epitopes, or you perform chromogenic labeling with alkaline phosphatase. So just don't take the first buffer that you find on the bench, or you borrow. There is a think it through what you are actually doing, what kind of assay you are performing, so you are using the correct one. And as I said, in some cases, the manufacturer might specifically recommend one buffer over the other PBS T or TBS buffer. Please follow their instructions. If you don't have any particular reason why you alter the protocol, but then something that you probably validated before that it works. Just a few more. I still have 4 minutes, so just a few words. Then it comes to the large samples. We will have a whole talk after me, very nice by David. But I just want you to remember a few things that it comes to the large samples. They are large, as the name. It can be intact organs or large organoids or very thick tissue, and they actually require extra attention before, during, and after the immunostaining procedure. I will, and then when we talk about the immunostaining procedure, I will focus a little bit about the distribution of your staining through the sample. You have a big sample, you have an organoid. What you want is to be sure that all the antibodies are penetrated and reach their target even deep inside of your sample. So it's very critical. Then you acquire images to pay attention and see. Ask yourself, I don't see anything. I went through my sample in the middle. I didn't really see any staining. Well, ask yourself, can it be biology or something else? It is something that you expect that it will happen with your sample. Maybe only are certain cells that are the outer part of your organoid. But sometimes you say, no, it should be all through. Then start to think, why don't I see anything there? Or why is my nuclear staining weaker in the middle of my sample? It cannot be. They should be the same everywhere. Or why is my staining always so bright at the outskirts of my sample? And here I have an example. I got it from my colleague, who actually also pointed exactly this out in some samples. They stain with 2 different antibodies, and you can very nicely see that the outer part of this organoid, it shows a very bright signal with both antibodies, but nothing in the middle of the organoid. And there are cells, so you cannot say, oh, maybe there is no cell, it's an empty hole, or I don't know what, because the cells also endogenously express GFP. So definitely, this means that there are some antibody penetration issues here, and you have to deal with them. Why is this happening? Is because quite often the antibody will bound to the first encounter epitope, so it's not too much left to diffuse further through the sample. Or some sample, they have a very dense extracellular matrix that will be like a hinder, or will hinder the antibody to penetrate and further into the sample. But there is nothing to despair about it. And other people also had these problems. So there are a lot of protocols available to optimize this issue of antibody penetration in thick samples. You just have to find the one that works for you, for the size of sample you are working with, and of course what type of clearing you are using, because obviously if we have this big sample, we need to also clear the sample and be able to look through. And here I just provide a few examples of protocols that we deal exactly with how to help the penetration of the antibody deep into the samples. Thank you for your attention. Questions, please. I think I was almost spotting time.
Yeah. Let's see. Your questions.
I have a question regarding the antigen retrieval, because we do the PFA fixation to cross-link. No, like during PFA fixation, we cross-link the sample, and this improves the preservation of the tissues. But then in antigen retrieval, we break part of the cross-linking. Is this not causing that we preserve worse the sample?
No, because that's already done. So, I mean, it's not always necessary to perform antigen retrieval. For example, if you have monolayers of cells, or it's not always necessary, and it's much more an issue when you have big samples like tissues. You have a block of tissue that will take quite a lot of time to fix, can from hours to days. And then you are also embedding it in paraffin. So this is when sample is already fixed, but afterwards some of the epitopes, because what might be hidden due to this long fixation, which you actually need, because if you don't do your tissue will go bad. And then you want to remove some of the bindings, that they were formed to be able to unmask the epitope. But after that, once the sample is fixed, and if it's fixed properly, then you shouldn't have a problem. The problem is you overfixed, which can happen very easily, especially with big samples, that you can overthink, and certain the outer parts of the sample might be more fixed than the inner part of the sample. If you don't have a special method that you have a more even distribution of your fixative during the whole fixation process. So no, the fact that you fix, it's an important step. It has to be done. Nothing, don't do light imaging, but after that, so if you adjust the time, height, well, then you shouldn't have an issue with or the fact that you need to do antigen retrieval. Not always necessary cryosections or certain methods, then you prepare your cryosection or the cells. In general, you don't need to do antigen retrieval.
Do you do antigen retrieval with your samples that are not preserved in that are cryosectioned, or what do you?
I don't do antigen retrieval,
Okay.
but I don't embed them.
All
It's
right. Yes, exactly.
whole mount. I fix in PFA, but I don't embed them in
paraffin.
resin.
Yeah. More questions?
Yeah, I have a question about the blocking part. In the slide, it shows the primary antibody is from mouse, and it enters the mouse antigen. I would like to know how this happens. You made something, changed something in the antigen, or how did you make this happen?
On the blocking?
Yes. Yeah,
So-
it's the slide. Before you use a secondary antibody, you block the
Yeah, so-
antigens. Yeah.
Yeah, so basically, you will get an FAB fragment that will bind, basically, to recognize these sites.
You're not sharing, Abby.
Oh, sorry. Okay.